All right. Yesterday, we had been talking about the first stage in cell respiration. And one of the things that I did not emphasize, or I think even mention with you all yesterday, was that this whole process that we're going through is taking place in what type of cells? Eukaryotic. Good. It's not happening in prokaryotes because we don't have that mitochondria. So if we're looking at aerobic respiration, we're looking at a eukaryotic cell. We're looking at mitochondria. It has to be eukaryote, can't be a prokaryote. We started off talking about the first stage. The first stage does not require oxygen. So glycolysis does not require oxygen. It is oxygen free. And that's important because when we're looking at other cells that are not eukaryotes but are prokaryotes, we do see that they perform glycolysis because they don't need any organelles, and you don't even need oxygen to do glycolysis. So bacteria, for example, will perform glycolysis. What's that? They will not do the rest of it. So they will not do the pyruvate oxidation or citric acid or the oxidative phosphorylation. But they will do glycolysis. And so will every other living thing on the planet. So one of the things that we have in common with prokaryotes and everything else, every living thing, is that we all perform glycolysis. So because of that, researchers hypothesize that the oldest stage of this whole respiration process is glycolysis. Because from the most simple bacterium to the most complex organism that you have, everyone performs glycolysis. Okay? It doesn't use oxygen. Back a long time ago, when the Earth was first formed, there was no oxygen. There was no free oxygen. So you couldn't perform aerobic respiration. You had to do something a little bit different, which included glycolysis. Okay, so that's an important point to remember for down the road. We left off, we had done glycolysis, and we had then talked about the oxidation of pyruvate. Remember, pyruvate is our 3-carbon molecule here. And it is going to have a couple of electrons liberated. It's going to have a carbon dioxide removed. That we're going to breathe out. That's the carbon dioxide. That's part of the carbon dioxide we breathe out. And it's also going to transfer some electrons to NAD to become NADH. And then it will combine with this molecule called coenzyme A to become acetyl-CoA. That step, oxidation of pyruvate, has to happen in order for the rest of this whole cycle this whole process to happen. Anybody remember what the next one is? Citric acid cycle, yeah. So we're going to talk next about citric acid cycle. And there's some slides in between here and the next photo, but I'm going to go to the picture that's from your textbook. This one here. So here I have my pyruvate being oxidized up at the top. Right here. And they end up with this thing, acetyl-CoA. This is called citric acid cycle that we're going to focus on. It's also called, you sometimes hear the word Krebs. It's named after Krebs, the person who first figured out all the intermediates and all the parts to the Krebs cycle. Krebs cycle will only happen if you have acetyl-CoA. So once you make that acetyl-CoA, then there's going to be another series of reactions that occur in Krebs cycle or citric acid cycle. We're only going to be interested in a few of those details. We're going to be interested, and again, numbers are not really what we're focusing on. We're not focusing on the number of ATPs produced, the number of NADHs produced. We're just focused on what is being produced, okay, not the actual numbers. So we're going to produce, it's kind of hard to see these, so I'll make them a little bigger. We're going to make carbon dioxide. We're also going to breathe that out. We're going to transfer from our acetyl-CoA. That's what's left of our glucose. We've still got a little bit of our glucose, a couple of carbons from our glucose left. We're going to strip that molecule of the rest of those electrons. And those electrons are going to be transferred to two shuttle carriers. One you're already familiar with, and that is our NADH. So NAD gets reduced again to NADH. So now this NADH is carrying more electrons from the remaining carbons that we had from the pyruvate, or from the glucose, actually. We're also going to make a little bit of ATP. If I make it directly like this, what kind of phosphorylation is that? Substrate level phosphorylation. We immediately transfer a phosphate group onto the ADP to get ATP. Now, we talked about NADH. We talked about how it's a taxi. It's a carrier of electrons. It's a shuttle. Just when you thought it couldn't get any harder. FADH2 is another shuttle molecule. It's going to pick up some more electrons. I will explain the difference between, well, I can explain it really now, I guess. NADH and FADH2 obviously are different chemicals. They're different molecules. But the big difference is these two carriers of electrons are going to generate a significant amount of ATP that we haven't generated yet. So far, we've just been using substrate level phosphorylation. We've only generated a little bit of ATP. We are going to take those NADHs and those FADH2s, both of those things, and we're going to move those to the last part of the aerobic respiration process. And those will generate ATP. The difference, functionally, the difference between NADH and FADH2 is one can produce more ATP than the other. And that's this guy here. NADH is the winner. That molecule can produce more ATP from the electrons that it's carrying than FADH2 can. So, but it's really not a very important point for us. They both carry electrons. They both take them to the same place. It's just one is a little more energetic than the other one. Yeah.
But why would there, what's the scenario where they would have, where the cell would rather have that ATP?
I don't think they actually know why there's two carriers and not just one. Okay. The hypothesis is that these are a series of reactions that developed over millions, hundreds of millions of years. And it may be that at one point they started running, the environment started running low on the precursor for NADH. And developed a second energy carrier, the FADH2. But no one really knows why. There isn't, it's not, they're both used all the time. It's not like one is used preferentially. Maybe FADH2 just became, maybe they weren't able to, the cells weren't able to use all of the electrons that were being stripped away, and maybe FADH2 came in and was able to do that. And that's like chemical evolution. And I don't know if there's really, I don't know what the hypothesis is for sure about how that evolved, but it's not like one is used sometimes and the other one's used other times. They're both used all the time. All right, so let me just take you back to the writing. The writing is less important, I think, than the picture is. The citric acid cycle is also known as Krebs, and it completely breaks down the pyruvate that you have. And so all of those carbons are now going to be liberated away from that glucose molecule that you started with. It became pyruvate, that became acetyl-CoA. And all of those carbon molecules will be given off, and we will lose that those carbons as carbon dioxide. In this cycle, you generate ATP, NADH, and FADH2 per turn. And we say per turn because how many acetyl-CoAs went in? How many started with one glucose? You had one glucose. And you made that into what? Actually, I'm just going to go back here. I'm going to erase that. I'm just going to have one line. One glucose became how many pyruvates? 2. So 2 pyruvates becomes 2 acetyl-CoAs. And so if you're doing the cycle, each one of those acetyl-CoAs has to go through the cycle. So when we say one turn, that would be getting rid of one of these guys. We have to do it 2 to get rid of both acetyl-CoAs. Okay. One time, one turn. Second time, 2 turns. That pointer really doesn't do a very good job. That's supposed to be acetyl-CoA. So we're actually making we're making 2 ATPs, 6 NADHs, and 2 FADH2s. But again, we're not focusing on the numbers, but we've got to go through this twice. And in the end, you're going to regenerate the molecule that went into the cycle. So if I look at, my molecule, let me erase some of this stuff here. Actually, they don't even show it on this one. There's a molecule that goes in here, and it joins with acetyl-CoA, and it comes back and it gets regenerated again. It goes into the cycle again, over and over again. So it gets reused. Let's see, what are the words on this other slide here? There's 8 steps, so there's 8 reactions. Each has a different enzyme. You don't need to worry about those enzymes. They tell you my biochemistry story when I took biochemistry at Northwestern. So I took biochem as a grad student at Northwestern, and it was a course that was for undergraduate pre-med majors, and the professor was a retiring professor, and the test we had on cell respiration, we had to know every reaction. You know, there's like 40 reactions, and you had to know all of them. It was open book, open note, but it was also timed. And it was so hard that he told us, and I guess I didn't pay enough attention, that he says, you can't use your notes, even though it was open. It was so hard. You had to have everything memorized, because if you had to look something up you wouldn't have enough time to get everything done. So on the first test, the average for the class was a 30%, and I got a whopping 15%. Yeah. Whenever I see there's 8 steps, there's like 16 and maybe 12 in glycolysis, I was like, oh my God, thank God you don't have to memorize all those steps. Because then you had to, you had to know exactly where the carbons were and how they were attached to it was awful. It was awful. There's a lot of gobbledygook here in this slide that we're not really going to be worried about. Here's the intermediate that I was talking about that comes oxaloacetate. It took me a long time to be able to say that word. And it forms when oxaloacetate and acetyl-CoA form together. The first thing that they form is citrate, and that's where citric acid cycle comes from. But we're really not worried about that. We're worried about the fact that what we make. So we're making ATP, we're making NADH, we're making FADH2, and we're making carbon dioxide. The last thing we need to know is this is taking place in the mitochondria. Okay? It's no longer in the cytoplasm. Now we're in the mitochondria. Those are the key features that you need to know about that. I mentioned all this with the whole acetyl-CoA and the oxaloacetate, but you don't need to know that. What comes into the reaction? Acetyl-CoA. What happens to it? You get ATP, NADH, FADH2, and CO2. And where does it take place? Mitochondria. That's what's important. Okay? Say it again. Mm-hmm. Yeah, it needs a little bit of jump start to get there. So it'll use energy that it already got from another reaction to jumpstart the breakdown of glucose. So 9.4. Whew. I think we got time to do this. This is a hard one. No, we got too much time. We got to do it. So far we've generated pretty little in terms of ATP. Very few ATP molecules. And it's all been done by that substrate level phosphorylation. So we're taking that phosphate and putting it onto our ADP in one fell swoop, in one reaction. Most of our ATP is not made that way. It is made through a process called oxidative phosphorylation, and along with or in conjunction with something called chemiosmosis and the electron transport chain. So just when you, again, thought it couldn't get any harder, sorry. Following glycolysis and citric acid cycle, NADH and FADH2, that's where most of our energy is. That's where all those electrons are. And those electrons are high energy. They have a lot of energy in them. And that is going to be used to produce the bulk of our ATP. And that is going to happen in our third and final stage of aerobic cellular respiration, okay, called oxidative phosphorylation. Come on. Okay, so we have to talk a little bit about the structure of the mitochondria. Or mitochondrion is a singular term for mitochondria, which is plural. The electron transport chain, where all of this is taking place, is located in the inner membrane or the cristae of the mitochondrion. So that is this guy here. That's my inner membrane, kind of poorly drawn. And notice how it's folded. Why might it be folded? more surface area. So that more surface area is important because embedded in that membrane are all sorts of little proteins. And because you have more surface area, you can have more proteins embedded in there. And those proteins are going to be part of this whole process. This whole slide is kind of an overview. And I want to talk in more detail about each step. I think, again, the pictures are probably more helpful. This is from your textbook. This whole piece in here is a phospholipid bilayer. That is the cristae or the inner membrane. Okay. Inner membrane or the cristae, same thing. And those little red pictures that I just showed you on the last page, where was that? Was it there? No. Yeah. Those little red pictures are actually not going to be red because whenever it's a textbook, we're going to represent them as purple. So those are our proteins. Yep. So all these purple embedded pieces here. Oh, that's not what I wanted. Ooh, let's clear that out. What? Oh, that just did pen. Why? Okay. There's a protein, there's a protein, there's a protein, and there's another protein. Okay, these are all proteins. They're all embedded in the mitochondrial inner membrane. Makani. Yeah, this guy here. So that kind of pinkish red, that's your phospholipid bilayer. And that's where all of these proteins are stuck, embedded in the membrane. Let's see if I got another. Here's another picture of it. Now they've actually just reversed it. Again, I have, this is my membrane. That's my cristae. And these guys here are my proteins. Okay, anatomy wise? Any questions? Any other questions? I don't know why I have nothing there. That's interesting. Okay. So I'm looking for the best picture to show you all this gobbledygook. Those are the same pictures, aren't they? Oh, this one's got. All right, let's start off with this guy here. I'm going to erase this. Our NADHs and our FADH2s, are carrying electrons. But they are only taxis. They're just delivering them. They're delivering them to this point. So they're coming from glycolysis that's happening in the cytoplasm. They're coming from another part of actually from the matrix of the mitochondria, if it's talking about citric acid cycle. And they're dumping them here. They leave them at this point. If it's NADH, it's going to drop it off and it's going to become NAD again. It's going to drop it off at this protein complex. But it's going to drop it to a carrier. So my electron gets transferred here. I got a lot of lines. Let's get rid of that. My electron's going to go to a carrier. Another carrier, another taxi. But this taxi is going to be a short one. The process of taking that electron from the NADH and giving it to another carrier releases a small amount of energy. That small amount of energy is going to be used to pump a hydrogen ion out of the inside of the mitochondria. That electron still has energy. And that carrier is like, yep, I'm done with you. Passes it to another carrier. As it passes over that protein, that second protein, another little bit of energy gets released.
So does it interact with the protein now?
In that, the protein is capturing that energy and it's using it to pump a proton again out
So against our concentration.
against this concentration gradient. Yeah, there's not a lot of H's down here.
Why is it pumping it up?
Well, we're getting to that.
Okay.
We're not there yet. It's going to do it again. At this point, the electron is pretty well spent. Notice how it's showing it going down. And that's done on purpose because now there's very little energy left in that electron. But there's still a little bit of energy to pump yet another hydrogen ion against this concentration gradient. One more time. This is active transport. We talked about this last unit. Takes energy. The energy is provided by the transfer of those electrons. As those electrons move across each one of those proteins, provides just a little bit of energy to move that hydrogen ion against this concentration gradient. Okay. In the end, the electron gets passed to oxygen and becomes water. Water. Becomes water. That is what we call the final electron acceptor. Oxygen is the final electron acceptor. All right, questions? I know it's a lot to take in. We will go over this again tomorrow and the next day. I'm thinking about giving you an Amoeba Sisters video.
Yeah.
You want to do Amoeba Sisters? Yeah. I think I will. Yeah, it's pretty good. All right. You confused about what?
What is the FADH?
Okay, that was going to be my next. I wanted to make sure that you were mostly okay with what we've done so far.
Where does the NADH go and why is it turned into NAD?
So the NAD, you're liberating electrons and you're liberating a proton, and that proton's getting pumped out. The electrons are getting transferred.
So are those what losing the protons? So the electrons in FADH are going into FAD,
Yes.
and that's what makes it FAD. Okay.
Yeah.
And
So-
then the protons going into that little protein thing.
And they're getting pumped out here. So we got 1, 2, 3. How many protons got pumped with NADH?
Probably
3. Notice FADH2. Where does it start?
second.
It starts with the second protein. So how many H pluses can it pump? 2. NADH can pump 3 hydrogen ions. FADH2 can only pump 2.
Wait.
Remember how I said that NADH is a little more energetic, a little more powerful than FADH2? This is why. This is called the electron transport chain. This word here, electron transport chain. This is half of oxidative phosphorylation. Wait, what? There's more? Have we made any ATP yet? No, we haven't.
Wait, how can you make?
Tell your cells that.
How can you make ATP when like the electron doesn't?
Okay, wait, that's a good question. So Tyler's question is, how can you make ATP if the electrons don't have any energy anymore? So where's now the energy kind of being, quote, stored?
The water?
Not in the water.
Oxygen?
No. What did we spend so much time doing? Transferring.
Oh.
We were using all this force to take the hydrogen ions and put them where they don't want to be. There's a lot of H pluses out here, and we just continue to pump them out here. They don't want to be there. Where would they like to be? They would like to be on the other side. All right. So this is electron transport chain. Oh, we got some stuff that wants to stay up there. Let's look at this. So let's look at the picture first. I think that will be more helpful. Let me get rid of those though. Okay, same picture that you saw before, but now we've got a fourth protein here. See the fourth protein on the end? Wait, I'm so confused. It's okay if you're confused. It can only do two. Yes, you're correct. Here's our fourth protein. This protein is called ATP synthase. Ooh, what do you suppose that does?
Does it make ATP?
It makes ATP, yeah.
Why is it?
This is so dumb.
Why is it that shape? Why is it a peripheral?
Yeah, it is. It's integral, so it goes all the way through the membrane. Because what's going to happen is water in a dam. You get a little crack in the dam. Where's the water going to go? It's going to go through the crack, right? It's going to. You got this big wall. You got a little crack. Where's the water going to go? Into the little crack, right? There's only one spot in this membrane where hydrogen can get back across the membrane, back to where it would like to be. And that is through this protein, ATP synthase. So they kind of queue up waiting to go through this protein. And there's not just one, there's, millions of them in the membrane.
Is it an enzyme?
It is an enzyme, yes. And it's going to catalyze the reaction of ADP into ATP. So now these guys are going to make a beeline for this protein. It is a race. And as they get down there, there's actually, I hope I have a picture of this. I used to have a picture of this. Yeah. I'm going to go to this next picture. Here's another picture of that same protein. As the H pluses, so these are my H pluses. They're still out here. As they go through this protein, they kind of spin the protein like a water wheel.
How do they know?
Magic.
Come on.
No.
No microscope is that good.
They do cell fractionations, so that's part of it, where they actually freeze the cell and then cut it open, and then look at it under a very high-powered microscope. And then some of it is guesswork too. So it flows through there, and you've seen a water wheel, right? Right. I mean, if water flows over a wheel, the wheel will start to turn, right? And then you can actually use that. In the old days, they used to use that to grind grain to make flour. That was how they did it back in the day. They used the energy from the flow of the water down its concentration gradient, or down from gravity, really, to cause the wheel to spin. That caused the grinder to grind, and you would get flour out of it. We're not making flour, we're making ATP. Okay. So as it flows down its concentration gradient, it spins that thing, creating energy that can be used to phosphorylate. Yes, kinetic energy. Good. That can be used to phosphorylate ADP into ATP. Okay. That's oxidative phosphorylation. And I think that's actually shown in the previous page. This guy here. Yeah. Oxidative phosphorylation. You still don't buy it?
I just don't buy it. It's like dinosaurs. It's like, come on, really? I work at a museum. My job is.
Where do you work? Do you really?
Yeah.
Wow, that's a cool gig. How'd you get that?
I just said it.
That's really cool. Very cool. So you don't believe in dinosaurs then? Are we done?
It's like,
We're never done.
It's like, really?
All right, so I think you're probably full up. Are you full up? Yeah.